Hi biologists, let's talk about how to do gel electrophoresis. I'm going to walk you through every step using the outline that is in your lab handout. And so please follow along with me in your lab handout and take notes on your lab handout with tips and tricks as I go through the lab. So the first thing that you have to do in gel electrophoresis is you have to do what we call casting a gel. And that's actually making the gel that you are going to use to run your DNA through. So how do you cast a gel? You're going to use what's called a casting stand in order to do that, and here is what it looks like. It has a lid, it's got these different plastic pieces inside, and you're going to move them around in order to help you do this. So the first thing it tells you to do is to place this on a level surface. I have to hold it up so that you can see. And you're going to place the gel trays into the two cavities. So these will already be there for you. The gel trays are these plastic trays, and they go here inside the cavities or the spaces in the tray. It says to make sure that the straight edge is on the right side. So I'm going to spin this to face me, and there's a straight edge, and it's on my right. The next thing it tells me to do is to insert the comb. This is a comb right here. The comb with the slots at the top of the casting stand with the 6 well side facing down. So if you look at this, you'll see that the comb has 2 sides. It has a 6 comb side with 6 bumps, and it has a 9 comb side that has 9 bumps on it. The directions tell me to have the 6 comb side facing down. So that's what I'm going to do. And I'm going to put it into, there's these little slots on the side, and I'm going to put it into those slots. And there's only one way it fits, so I have to put it like this. So I'm slotting it into there, and so it will stand up on its own if you've done it correctly. And once again, the 9 bumps, the 9 wells should be on the top, the 6 bumps should be on the bottom. So that's my first step. The next thing that's going to happen, I'm going to do for you. It says to partially peel the film of a gel green cup and microwave for 20 seconds. Allow it to cool for 15 seconds and don't microwave more than 5 gel cups at a time. I will microwave this for you and hand it to you when you walk into class. So when you get it from me, this cup will be very, very hot. Let me show you what it looks like. So it's full of this liquid right here, and this is what's going to make our gel. And so I've boiled this in the microwave for you, and I'm going to hand it to you once again. It's very hot, so be careful. And the next thing you're going to do is you are going to use this gel cup to make your agarose gel. So it says to slowly pour this hot agarose solution into a gel tray. Make sure there are no air bubbles in this agarose solution. Let the agarose gel solidify for 10 minutes or until opaque. Do not disturb the gel until time is up. So I'm going to carefully peel it back. It will already be open, so you just have to kind of re-peel it. And once again, I'm only going to pour it onto one side. So pick the right or the left, pick your favorite side. I'm going to pour it onto one side. And I'm going to pour it really slowly so there's no air bubbles. So let me see if I can do this for you. So I'm going to, got my cup. And then I am slowly just going to pour it in here to my tray. And then I'm just going to let it sit on my table for 10 full minutes without disturbing it. I'll see you again when 10 minutes is up. All right, my 10 minutes is up, so now I'm ready to move on to my next step, which is removing the comb. So it says, carefully remove the comb when the gel is ready. Remove gel tray with solidified gel from the casting stand and wipe off any excess agarose from the bottom of the tray. So my gel is really firm. I can see it looks firm in my tray. And so I'm going to take the comb out. And when I take the comb out, I want to make sure I pull it directly up so that I don't break the gel or ruin it in any way. So I'm going to take this and very carefully, if I were you, I'd be using 2 hands. I'm going to carefully pull up from the gel. That went really well. So now I've got my comb. It's free. I can put it to the side. And then I'm going to take the actual gel out of the casting stand. So this side has my gel. I'm going to put my fingers on both sides of the plastic casting tray and just kind of gently wiggle it out. Sometimes there can be gel that got onto the bottom of the tray. This happened to me a little bit, so I'm just going to take a napkin and carefully wipe off anything that's on the bottom right here. And then we're good to go for our next step. So our next step is actually putting our gel into our tray, into our gel electrophoresis machine to run our gel. So the next thing it says is to ensure the black viewing platform is in the tank if it's not already installed, and put the gel along with the gel tray into the tank. So this gel is going to stay inside of this plastic tray the entire time. And to make sure that my wells, see if you can see them, they're the squares that we're going to put the DNA into here at the top, that those wells are aligned with the marks on the platform on the negative end. So let's see what they're talking about. So today we're going to be using, this is the tank from our gel electrophoresis system. It's clear and plastic, and everything that happens today is going to happen here inside this tank. This tank fits inside of the gel electrophoresis power station, which is black plastic and looks like this. We don't want anything to happen in this. This is solely a power source. So no liquid should go in here, no DNA, nothing should happen in here except this tank fits really nicely inside. So once again, we're gonna put things in the tank separately right now. So I've got my tank, and you can see that there should be this black plastic piece inside the tank that has outlines of squares that are my wells. So I'm going to take my gel and I'm going to insert it into my tank, ensuring that my wells are on the same side, and it will just slide right in. So now my gel is on top of that, ready for me. Okay, let's see what we're gonna do next. So next, we're going to measure our buffer and put it into the tank. So at this point, if your tank is out like mine is, you're going to actually put your tank inside of the power supply and make sure that it snaps fully into place. Right now, I'm gonna change the view of my camera so you can see what I'm doing next with the running buffer. All right, so this is what the gel looks like from above. The next step tells us to measure 135 milliliters of running buffer and pour it into one side of the tank to push out the air. There will be running buffer available in beakers. I've already measured out 135 milliliters here in my graduated cylinder. So I'm going to pour this buffer into the tank on one side only. So I'm going to do it on the positive side. I'm going to slowly pour it in, and I'm going to pour it all in, all 135 milliliters, all on one side of the tank, and you will see that it will fill everything up. It should flood the entire tank like that if I have done it correctly. Okay, there should be a nice even layer. There should be no air bubbles if everything has gone right. All right, the next thing that we are going to do is we are going to place the power supply into the wall and put the tank into the carriage. We actually already put our tank here in the carriage, so it's nice and snapped in. I think it's better to do that before we pour our buffer. So we've got that all in, so now we're gonna plug it in. So you have a power supply that plugs, one side plugs here into the back of the machine. The other side goes into the wall over here. Okay, and then we're ready to go and to start loading our gel using our micropipette. So the next thing we're going to do is, it says to turn on the low intensity blue light by pressing the button on the carriage to help visualize the wells while loading. So there's a high intensity light right here and a low intensity light on the bottom. We're going to do the low intensity light, the second button right here. We are also going to load 10 microliters of DNA per well. So you can see here I've got my micropipette. It is set to load 10 microliters. So it's going to look like the number 100 on my micropipette. And I've already got my tip onto there. So remember, you should set your micropipette and then put the tip on. It tells us to remember to change tips between samples. That's going to be really important. And to load tips, to load the samples according to the chart. So you're going to have to look at your chart to figure out what order you're going to load the DNA in. It's in the directions to your lab. But for everyone, we're loading 10 microliters. So I've got my DNA sample right here. I'm going to use good pipetting technique, so I'm pressing to the soft stop, and then I'm going to place the tip of my pipette here into my DNA. I'm going to slowly release that, and if I've done it well, you should be able to see that I have a fully full tip there at the bottom of my pipette tip. Okay, now I'm going to do this one-handed. We'll see if this works. I'm aiming my pipette tip for the corner of the well. So I'm aiming it for right there, and I want to get absolutely all of the DNA inside of that well. So I'm going to place my pipette tip into the corner, I'm gonna slowly release all the way down and pull out. That wasn't so great. You could see there was kind of a cloud of DNA around that. So I'm gonna try again, but before I do my next well, I'm going to eject my pipette tip. So I'm gonna press this button here on the side, and I'm just gonna put it here on my lab table, take that tip off, and I'm going to get a new pipette tip. All right, I've got my next DNA sample loaded up. I'm gonna try and see if I can do this again. Once again, I'm aiming for the corner of the well, and I'm gonna release all my DNA. I'm gonna keep my thumb down until I pull it fully out. That was much better. You can see how in my first one where I didn't do a great job and it was really cloudy, there's not that much inside of the well, but in my second one, it looks nice and dark. That's how it should go if everything goes right. Once again, I'm gonna eject my pipette tip onto my lab table, and I'm gonna do my third one. Once again, I've got my third micropipette, fresh tip, full of 10 microliters. I'm aiming for the corner of the well. I'm slowly gonna eject everything. Not a great one, you can see a lot of clouds. I'm doing this one-handed, you'll use two. I'm gonna eject my last pipette tip, and then I will be ready to actually run my gel. So my samples are all loaded according to my chart. You will have maybe more samples than me, so make sure you're following your own directions for your lab. So once the gel is loaded, it tells me, please don't move it. Make sure my power supply is plugged into the wall, check. And place the photo hood, that's this orange hood, on top of the carriage. So it's gonna look like this on top. Okay. Turn on the unit by pressing the power button here in the corner. So I'm gonna press it, and if I've done everything right, the green LED light will turn on. If the green light doesn't turn on, please come get your teacher and we will troubleshoot and make sure that happens. All right. We are going to allow the gel to run for approximately 25 minutes. I'm using my phone right now, and if I place the camera to my phone directly over this hole in the photo hood, it is already set to be able to record a great picture or video of what's happening inside. You can see that at the top there's some air bubble, some air bubbles starting to form because the electrodes are working. As it goes, we'll be able to start to see the DNA move. You can already see there on the very left side of the screen some glowing bands. That is our DNA that is starting to move. So the directions tell us to let this run for 25 minutes. You can take photos and videos while it does so. And at the end, we will turn it off by pressing this button once again. And we'll actually turn on the bright light right here in order to see everything really, really well. Okay, so now we've done all of our gel electrophoresis. Once you've taken all your photos, you've gotten a great visual to hold on to and use to analyze your data, then we're going to need to clean up. So I've unplugged my gel electrophoresis unit. It looks like this, and now I'm gonna do my steps to clean up. So the first thing I'm gonna do is I'm going to take the orange hood off and just put it carefully to the side. And the next thing I have to do is out of my running carriage, the black thing, I need to carefully remove the clear plastic tank that is inside. Remember that this tank is full of the liquid running buffer, so I have to do that really carefully. I'm gonna take it out, so I've separated my tank, and I'm gonna make sure that there's no liquid at all inside of here, inside of the black carriage, and I will dry it off if it does. I've got my tank, it's full of liquid. I'm going to dump this running buffer down the drain. So I don't have a drain near me here in my office, so I'm just gonna dump it into this beaker instead. You will dump it down a sink, and you're just gonna carefully pour off all of the liquid, and make sure that the gel doesn't fall into the sink while you do it. Okay, so I've dumped all of that out, so that's good. Then I'm going to remove the plastic tray out of here that has the gel in it, and I'm gonna go and throw this gel into the trash can. So it's gonna go in the trash, and then you're going to bring your teacher both this plastic tank that was used to run our gel and the plastic tray that used to have the gel in it. You're gonna put the gel in the trash. That way they can rinse it off and clear it out so it's ready for the next class. All right, can't wait to see you for gel electrophoresis.